currently I'm an undergraduate student from Purvanchal University, Nepal. I'll be presenting two interconnected projects today. One called Reverse Central Dogma, and another, the current project that I'm working on, is Seed to Lead. These two interconnected projects, what they do is they challenge the long-standing paradigms in biology. So let me move forward. The first central dogma, the reverse central dogma, asks, can protein encode the nucleic acid sequence? Through the molecular docking simulation involving 12 distinct peptide molecules, we have found that there are specific recognition patterns among the amino acid and the nucleotides. So the next seed to lead uh, project aims to screen the natural lead driven product from the cannabis compounds such as lignanamides and screen them against the potent drugs against cancer, targeting the specifically the NAMPT enzyme that is involved in cancer metabolism for the NAD plus synthesis. So this two project they kind of breezes the theoretical innovation to the practical applications of such theoretical concepts. So moving forward, the first reverse central dogma, computational exploration of nucleic to protein to nucleic acid information flow. So central dogma says that every time among the living system, the information flows from the DNA to RNA to protein unidirectionally. But can they be can it be reversed backwards? That's the question that the project aim to answer. There are exceptions to central dogma, such as there are the cases of reverse transcription where the information flow from RNA to DNA, and some prion-based inheritance are some exceptions. So through molecular docking, we have identified specific amino acids such as arginine-rich peptide motifs so the significant binding affinity with the specific nucleotides such as guanine with the energy binding energy of negative 9.8 kilocalorie per mole, which suggests that the binding energy was statistically significant, which was proven by statistical modeling uh, by using ANOVA, and we are currently focused on developing the algorithm for the reverse translational uh, program that would translate the peptide sequence into the DNA. So, which would obviously um, be applied in different field of biology, such as we could predict the gene, we could synthesize the gene by utilizing the bidirectional circuits, and it will assist in drug design. And parallelly, we are currently working on the project called Seed to Lead, where we are uh, focused on extraction, phytochemical extraction, their uh, in vitro cytotoxicity testing, and in silico analog prediction or kind of inhibitor prediction, where we are targeting the compound called enzyme called NAMPT here. The NAMPT is the key enzyme that is involved in uh, metabolism of NAD plus biosynthesis. What NAD plus does in a cell is it is the key molecule that is uh, involved in doing sort of oxidation reduction reaction in the cell. So for a cancer cell to survive and proliferate indefinitely, the cell need to be furnishing the energy. For that, NAD plus play its roles. Besides this, NAD plus is crucial in mechanisms such as DNA repair and a lot of things that and the cell rely upon the NAD plus to mm, sustain and proliferate. So this enzyme, we are targeting this enzyme specifically because the potent inhibitor that were previously used, such as FK86, was failed due to the systemic toxicity it created. Rather than the moderate, we are proposing the moderate level of allostatic kind of inhibition, moderate inhibition, where the inhibition would be moderate, that it would be specific to cancerous cells. It would specifically inhibit the cancerous cell to grow, and it will kill due to energy starvation, but the normal cell will sustain. And theoretically, it has shown that the therapeutic index of the our proposed compound CSCO3, which was designed by modifying the compounds extracted from the seeds such as cannabicin B. We took the scaffold of the cannabicin B and we modulated. We added such structure that could enhance the admit properties of the uh, lead compound that we are proposing. So in we are currently on developing the future protocols. I'm and we are executing this project at final year thesis of my own. uh, and I like to invite every collaborators, every feedbacks. I welcome every feedbacks, every questions regarding the status of the project that I am involved with. I'm open for the queries. Thank you.